Hi, good morning, everyone. I'm Kiele. I'm the field application specialist in Apical Scientific. Okay, welcome and thank you for participating in today's webinar with the title, The Headmaster of High-Fidelity PCR Phusion Plus. So when I go through the slide, if you have any questions, you can write down in the chat box, okay? I will try my best to answer you after the slide sharing. But if I cannot answer you or I may need to have more information from you, maybe I will get back to you through email later, or if possible, you can also email me at support@epicalscientific.com. Okay, yeah. So not to delay, let me start the sharing. Okay, the slide sharing. Okay. So as the webinar title, today we will talk about high fidelity PCR. So I will go through a short intro on this, and then we'll talk more on the type of the DNA polymerase, then what make fusion DNA polymerase. Okay, after that we'll go into detail sharing on fusion polymerase and fusion plus DNA polymerase and what it can give you on your study. Okay. Okay, so what is high fidelity PCR? Okay, first we need to know what is fidelity means. Fidelity is, you can say, is also refer to accuracy in a template sequence which in which a DNA polymerase is replicates. Okay, then what is high fidelity PCR? Okay, high fidelity PCR, it is a type of PCR that is designed to minimize error during the DNA amplification. And high fidelity PCR, it has some point which refer to this type of PCR. Okay, first certainly is a high fidelity, so it's high accuracy. So it will have high accuracy as it is using the DNA polymerase with proofreading activity, which it will correct the mistake upon the DNA synthesis and result in very few or uh, no errors compared to standard tag DNA polymerase. Okay, and next is because this high proof reading DNA polymerase, it have the 3' to 5' exonuclease activity that allowed them to remove incorrectly incorporate nucleotides and also replace it with correct one. Okay, so with this, high fidelity PCR usually will have very low error rate, like 1 in 1000 kb or even lower. Okay, and to ensure best performance of the proofreading DNA polymerase, there is a need to optimize the reaction conditions of the PCR, especially the buffer system and also the temperature setting. Okay, so high fidelity PCR, it usually will have higher cost because the DNA polymerase is more special. Okay, and but it is actually crucial for some application that when the accuracy is very important. For example, like cloning, that you will need it for expression, protein expression later, so you cannot have one single base that is different. If you have one single base that is different, then later on your expression, the protein will be different. Okay, so it affects the protein directly. Okay, and yeah, there is also on mutagenesis study, also sequencing or NGS study. Okay. Okay, there are a few factors in PCR that will contribute to the high fidelity. Okay, first, okay, certainly is the DNA polymerase. So different DNA polymerase they have different level of fidelity. So later on, we'll talk about this. Okay. Another factor is the proofreading activity. Similarly, different DNA polymerase, they have possess different proofreading activity. Okay, the level will be different. Okay, we'll talk about it later also. Next is the enzyme. Okay, enzyme will usually work under certain condition. So DNA polymerase also the same. It have a specific reaction condition that um, it need to have this condition so that it can work optimally. Okay, especially the magnesium ion concentration in a reaction. So it will determine whether this DNA polymerase, this enzyme, it works well, okay, it works under the control, or it works crazily by adding errors into the extended fragment. Okay, yeah. And also another thing is the DNTP balance in a reaction. DNTP balance, it can also influence the fidelity. Imbalance different of the this DNTP amount, okay, it will lead into increase of error. Maybe if you have, let's say you have DGTP, amount of DGTP more, then it will have create more error that DNA polymerase will put in this DGTP. Okay, so this is something that about the DNTP valence. Okay, and then last one is the quality of the template. A pure DNA template, it can increase the fidelity of the PCR, but if there is presence of contaminants or the DNA already damaged or smearing, okay, it can also lead to error during the amplification. Okay, so among all these factors, you can see most of them, like the reaction condition, is also refer to the condition for the DNA polymerase. The NTP balance also refer to the DNA polymerase, where it will input more each of the DNTP, something like that. Okay, so you can see all of them is talking about the DNA polymerase. So that's why we all know that DNA polymerase play a very important role here. So let's understand more on DNA polymerase. Okay. Okay. What is DNA polymerase? DNA polymerase, everyone know, right? So it's an enzyme that help create new DNA molecule by adding nucleotides to a growing DNA strand. Okay, so its main function usually include the DNA synthesis, proofreading DNA, replicate and repair in cell. Okay, so all these function, main function in the cell is these three. DNA polymerase, it has two type of activity. One is the 5' to 3' polymerase activity. So it allow it to read the original DNA strand and add nucleotide from 5' to 3' basic and form a complementary strand. And this activity is also important in DNA replication and also DNA repair, where it will help in copy the DNA before cell division and also fix the damaged DNA in a cell. Okay. Other activity from DNA polymerase is called 3' to 5' exonuclease activity. Okay, this activity is very important because it allowed the DNA polymerase to check and then correct the error in the newly synthesized DNA to prevent any mutation that is happen upon the synthesis. And this activity is also called proofreading activity. Okay, so these two activity, they can also have some relate with the property of the DNA polymerase. Okay, these two activity, it gives this property in the DNA polymerase. So what is the property? One is the proofreading, another one is the processivity. Okay, so what is these two property? Let's talk about this. Okay, first one, the proofreading property of the DNA polymerase. It refers to the 3' to 5' exonuclease activity in a DNA polymerase. Exonuclease, it actually refers to an enzyme that able to cleave the phosphodiester bond in the DNA from N. Okay, so if the one that cleave from the middle is called endonuclease, if you forgot about exonuclease and endonuclease, this is the difference. Okay, exonuclease cleave from the end, endonuclease is in at the middle. Okay, so this DNA polymerase also possesses this activity where it cleaves the DNA, the mismatch at the 3' to 5' direction. Okay, yeah. So in a DNA polymerase here, okay, um, amplification, it happened at the 5' to 3' polymerase domain. Okay, when it actually found there is mismatch, then the DNA elongation will generally will pulse. Okay, so it will pulse there, and then this pulse, it will actually allow the melting of the end of the DNA strand. Okay, and with this it free the 3' end, and the 3' end that is carry the mismatch. Okay, with this it free the 3' end, and then it allow it to enter the 3' to 5' exonuclease domain in the DNA polymerase. Okay, this domain. Okay, so at this position, the activity 3' to 5' exonuclease activity will happen. So it will cleave away the mismatched nucleotide. Then the amplification will resume back at the 5' to 3' polymerase domain. So the synthesis of the DNA continue here. Okay, so this is how the proofreading in a DNA polymerase occur upon the amplification. Okay, now how about processivity of the DNA polymerase? In DNA replication, the 5' to 3' polymerase activity, it actually refer to the activity where the DNA polymerase adding the nucleotide from the 5' to 3' direction. Okay, while processivity, it actually means on the continuous of this DNA polymerase adding the DNA, the nucleotide from 5' to 3' direction until the entire strand is synthesis or until it encounter any obstacles. Okay, so the processivity of the DNA polymerase is referred to the number of the nucleotide that added to a growing DNA strand in one single binding event or before the DNA polymerase dissociate from the template strand. Okay, so different DNA polymerase, they have different level of processivity. The one that with high processivity, it will tend to give you higher affinity for their substrate and incorporate more nucleotide per binding events. So it means it will give you a longer PCR product and a more complete amplification. And with this means it's also means that it will give you higher yield of the desired amplification product. Okay. Okay, so there are two family of DNA polymerase, which they actually categorize based on their sequence homology, structure features, and also functional characteristic. Okay, so type A DNA polymerase, they are typically involved in DNA replication and repair. And this family of polymerase, they are low in fidelity, low in processivity. So in other terms, it will means that it is unable to generate long PCR product. Meanwhile, the family B, okay, DNA polymerase, they involved in DNA replication with high fidelity because they have the proofreading activity. Okay, so family from type B DNA polymerase, it can give you higher fidelity and it also have better processivity. So it means it is suitable for long product amplification. Okay, so from here you can see that type A DNA polymerase, they are suitable for routine PCR application where usually the product is around 500 to 1.5 kb, something like that. Okay, so example is the tag DNA polymerase. Okay, or maybe in a real-time PCR that is you no need to have a very long product. Real-time PCR will have a short product like 1.5 sorry 150 base pair, something like that only. Okay, so type B DNA polymerase, it is suitable for high fidelity PCR, for example the PFU DNA polymerase, or application which they need a fast PCR also. Okay. Okay. So let's talk about fusion. Okay. So fusion, it is you can say is a type B DNA polymerase, but with a certain level of modification. So it enhance um the fidelity of this DNA polymerase. Okay. Okay, so first, why we call it fusion? Because it is the DNA polymerase. It is the fuse of the DNA polymerase from a proofreading polymerase, where it is like a pyrococcus species like the proofreading DNA polymerase. Okay, and the fuse of this polymerase with a double-stranded DNA binding protein from SSO. Okay, this bacteria. Okay, so the fusion of these two, they found that it can help to increase processivity without compromising catalytic activity and enzyme stability. So it will show enhance in fidelity up to a certain level. Okay, and then with a better processivity, and it allows the amplification of long product up to 20 kb with high yield and resistant to PCR inhibitor also. Okay. Okay, so the more scientific fusion high fidelity DNA polymerase, it will actually introduce in 2003. So it provides 52 times higher accuracy, higher fidelity than the tag DNA polymerase, and 6 times accurate than the PFU DNA polymerase. Okay, where we determine this. Actually, this number is actually there is a study. So the researcher, they run a PCR with fusion DNA polymerase, and also with the PFU and tag DNA polymerase for a 581 base pair long DNA sequence. And later on, they want to use this for protein expression in E. coli. So you can see from the gel here, it represent in the journal. Okay, so this gel, it show fusion polymerase, it can give you a good single band with high intensity when compared with two others band where you can find tag that is some unspecific. Also PFU, it have some specific. Okay, so in this paper, the researcher mentioned that fusion high fidelity DNA polymerase, when used for DNA synthesis, has dramatically lower error rate compared to the other enzymes previously used for such application. So its fidelity is 2 to 6 times higher and approximately 50 times higher than the fidelity of PFU and also tag DNA polymerase correspondingly. Okay, yeah. So after the intro use of this fusion DNA polymerase by Thermo Scientific on 2003, they actually add many different variants and format of the fusion enzyme that allow um, the that that they add into the product family to allow um, the meets of different needs of application. Okay, for example, like the fusion hotstart DNA polymerase, it gives you more specific amplification. Fusion U is for DUTP PCR, and there is also one DNA polymerase that is added into this family that it possesses highest fidelity. So meet the Fusion Plus DNA polymerase. So this Fusion Plus DNA polymerase, it is the Headmaster for high fidelity DNA polymerase. Why? Because it has an outstanding fidelity, which over 100 times more fidelity as compared to tag DNA polymerase. And according to the more scientific study here, it shows side by side comparison of the a few high fidelity PCR enzyme on the market and using the NGS method with molecular barcodes. And you can see here is Fusion Plus. It has a most outstanding fidelity as compared to other. Okay. Okay, let's take a look at other enhanced property from this Fusion Plus DNA polymerase. Why we call it as a headmaster? Because it have many different enhanced property. Okay, so first thing, what you look for your study, if you are looking for accuracy or maybe you need copies of exact similar sequence from your gene of interest um, later on for cloning or for gene expression, or your sample may carry PCR inhibitor. Maybe you want to amplify DNA from soil or from food, okay? Or you may have a few genes, okay, in one study. So you want to PCR all them in one PCR, okay? But you found that you cannot well optimize them into the similar melting temperature, so you can only co-cycle it, or you may have to perform some special PCR that for some different application like the DUTP, like site-directed mutagenesis. Okay, so all this they can be solved with fusion plus DNA polymerase. Okay, how? Let me show you here. Okay, so as mentioned just now, fusion DNA polymerase, it has high fidelity. Okay, so fidelity of a DNA polymerase can be measured using different methods such as colony screening assay, sequencing, or next generation sequencing. And here is a test with colony screening. So the insert is of a 3 kb will generate by PCR using um, DNA polymerase, fusion DNA polymerase, um, and also tag DNA polymerase and also fusion. Okay, and from here they proceed to cloning and then colony screening. So they proceed to cloning and then continue with colony screening. Okay, so from the result, they found that the one the that carrying the PCR product using tag DNA polymerase, it can give an average of 2 error per clone, because tag DNA polymerase, it have around one, the error rate is 1 per 1 kb, 1 error per 1 kb. So here with the big size amplicon, it will give you more error. So you can find that there is actually 2 error per clone. Every clone also have error. While the one that is PCR with fusion DNA polymerase, it give a very less than 1 error in 25 clones. It means when you pick 25 clones, maybe only 1 clones that have 1 error inside. Okay, so here I want to highlight is actually this one is the comparison of fusion DNA polymerase, and I don't have the data comparison for fusion plus DNA polymerase, but fusion DNA polymerase, it have the fidelity of 52 times higher. So fusion plus, it may give you even better um, uh, result for this. Okay, so maybe you will have even lower error per clone for Fusion Plus. Okay, okay. Also, Fusion Plus is suitable to use for PCR with DNA template that carrying high inhibitor. So Fusion Plus, it has a formulated reaction buffer, which make it highly tolerant to PCR inhibitor. And here is the example. So you can see from the gel, um, the well here. Okay, so from the gel, the well number 1 is the PCR with pure DNA. There's no inhibitor added. So, and then the well number 2, it carry the humic acid. Okay, so the DNA, they mimic the inhibitor inside, so they added a certain amount of humic acid into the DNA and then use it for PCR. Okay, and um the number 3, well number 3 is the PCR with DNA that carry hemine. Hemine usually is from blood. Okay, and then this well number 4 is xylem. Xylem is usually from plant. Okay, plant contaminant. Okay, so you can see here Fusion has no problem in amplify all these sample, while the rest of the polymerase, they do not give a very good amplification. Okay, yeah, some will have a very faint amplification. Okay, so here, if you are working on sample with PCR inhibitor, for example, the DNA is extract from blood, from soil, from plant stool, or possible from food sample also. Fusion Plus will be the best choice for your PCR. Okay. Okay. So next is here is a very special feature for Fusion Plus DNA polymerase. It has a new buffer formulation which allowed the DNA polymerase to work in a universal annealing temperature of 60 degrees Celsius. Okay, so this means the annealing temperature can be set at 60 degrees Celsius for all target, regardless the difference in the calculated temperature of the primer. So as long as the primer design, it need to follow the general recommendation. So it means before you design your primer, you need to go read through this guideline from Fusion Plus, and then you design your primer. And then regardless the primer temperature, melting temperature. So usually when you design primer, you will check the melting temperature. So melting temperature may be different. So it also means that your calculated annealing temperature here, it will be different. Okay, so from here you can see different calculated annealing temperature. Here has 59 up to 69. Okay, but this one is a single annealing PCR by using this temperature, the calculated annealing temperature. And here, this one in the left side, the gel, it refer to the PCR using Fusion Plus, and even though it's different calculated annealing temperature, but we are using one universal annealing temperature for all this, okay, at 60 degrees Celsius. And you can see the comparison. The amplification is still very good and consistent from here. Okay, even though they are using one annealing temperature only. Okay, yeah. Okay, so with that means you can co-cycle your PCR under a sample PCR, one under a same PCR protocol. Okay, for example, if you have here, like here, you have five different target which should amplify with five different primer set by using five different annealing temperature. So it means you will need to run one, two, three, four, five times of PCR, and it will take you 5 hours, around 5 hours or more. Okay, but if you're using Fusion, you can co-cycle, put all this PCR product in one plate, and then you set your PCR run by at the 60 degrees Celsius, and then you can done all this in one PCR, okay, using one same protocol, and it means you can done all this in around 1 hour. Okay, yeah. So this property is only found in Fusion Plus, and you can see from this gel, even under a cold cycle, the amplification is consistent and the yield is also as good as the one that is here, which is a single cycle. Okay, here is single cycle and here is cold cycle, and the result is about the same. Yeah. Okay, another property for Fusion Plus is it is suitable for GC rich PCR. Typically, GC rich, it means that the DNA region have a GC content of 60% and above, and this will mean that it has a higher melting temperature. It has a higher possibility of secondary structure formation, and all this, it will reduce the amplification efficiency in a PCR. So by using Fusion Plus, you can actually include a new Fusion GC enhancer, and it will give you high efficiency in GC rich amplification. For example, in this gel here, it's showing 16 targets with different GC content. Okay, so this show the GC content here. Okay, so you can see that Fusion Plus, it can amplify very well and produce the PCR product with high yield. Even the GC content is as high as 80%. Okay, and if compared to the other Fusion, okay, they are lesser in the performance. Okay. Okay, so as mentioned just now, there are many family members under Thermo Scientific Fusion DNA Polymerase family. So you can find them here. We have Fusion High Fidelity DNA Polymerase. We have Fusion Hostat 2 DNA polymerase, we have Fusion Plus DNA polymerase, and you can require for single polymerase, master mix, or the one that is come with green buffer. This green buffer, it carry the tracking die, and it allowed for direct loading for gel quantification. Okay, now how to choose. Yeah, how to choose. So here is our recommendation. If you have many genes to study in once and you don't want to waste your time to optimize each of them, then you can use Fusion Plus DNA Polymerase. So it can allow you to co-cycle all your target all your PCR in one PCR run. Okay. If you are working on high fidelity PCR for cloning, for gene expression sequencing or mutagenesis study, you can use the Fusion high fidelity DNA polymerase. If you find your PCR showing many different bands, okay, you can use the Fusion hot start to get the specific amplification for your PCR, and in the meanwhile get the high fidelity also. Okay, and also this can be used for multiplex PCR. And if you want to amplify long PCR product, you can use Fusion Flash High Fidelity PCR Master Mix. It has a best speed, so you can see here the speed of this Fusion Flash is the fastest one. Okay, yeah, so it have a best speed in amplification, so it will save your time for long PCR product. Okay, and if you're working on some special PCR to incorporate the NTP, okay, so you can use Fusion U, okay, hot start DNA polymerase. Yeah. Okay, so as a conclusion, what you take home from this webinar? Okay, first, what is a high fidelity PCR? High fidelity PCR, uh, it refers to high accuracy in the sequence as it is using DNA polymerase which carrying proofreading activity, and the error rate should be as low as 1 in 1000 kb or even lower. Okay, yeah. So what contribute to the high fidelity in the PCR? Typically, the property of the DNA polymerase and with the additional help from a well-optimized reaction buffer and DNTP amount. Okay, another one. What are the properties from the DNA polymerase that play the important role in high fidelity PCR? What is the property? That is two. One is the proofreading, another one is processivity. Okay, proofreading, it comes from the 3' to 5' exonuclease activity of the DNA polymerase, and it helps to correct mismatch upon amplification. Processivity refer to the number of nucleotide added to a growing DNA strand in 5' to 3' direction upon one single binding of the DNA polymerase. Okay, yeah. Next is what are the two major family that of the DNA polymerase. So as we discussed just now, there is type A and type B. So type A polymerase, it have low fidelity, low processivity. For example, tag DNA polymerase, so it should suitable for routine PCR. While type B, it have a high fidelity and high processivity, so it can amplify long PCR product. Okay, yeah. So what is fusion DNA polymerase? Is you can say it's a type B DNA polymerase. Is a modified um, DNA polymerase that fuse the PFU light proofreading polymerase with the double-stranded DNA binding protein from SSO. Okay, so it will give you enhanced property in both proofreading and processivity. So thermoscientific fusion DNA polymerase, it has at least 25 times higher in accuracy when compared with DNA polymerase. And one of the family member, which is the Fusion Plus DNA polymerase, it can have higher than 100 times fidelity as compared to tag DNA polymerase. And this Fusion Plus, it has some enhanced property where you can use it for co-cycling. You can use it for um, sample DNA with high PCR inhibitor, or you can use it for GC-rich amplification. Okay, yeah. So the fusion DNA polymerase, they can be used for many different application. Okay, which need for the high fidelity PCR. Okay, yeah. So with that, come to the end of my sharing. Thank you for participating in our webinar.